Thank you, Alex or Alejandro, for asking me to be a part of this. I'm going to talk about my favorite group, the Aphonopelma, the tarantulas of North America, or at least the United States. So first off, it's a chokma. So Holchepot Chris Hamilton, Chikasha Saya. And that is a traditional greeting from my tribe saying that my name and that I am Chickasaw. How are these? Oh, there. I'm trying to figure out how these slides will change. Okay. So this work is myself and Brent Hendrickson. It's kind of a continuation of our work when we started to revise the Aphonopelma here in the United States, and now we're expanding that into the Mexico side of the border, because we know spiders don't believe in walls. So I also have to introduce one of my students. I finally have some PhD students. This is Karina Silvestre Bringas. She's a new PhD student in the lab. She comes to us from Marshall Hadeen's lab at San Diego State, where she worked on Habernatus. She is an indigenous scholar, and one of the things that we're interested in working on is integrating indigenous knowledge or traditional ecological knowledge into our understanding of life history differences between these species of Aphonopelma and the Sky Islands. I also have to acknowledge that the University of Idaho is located on the lands of the Nez Perce and Coeur d'Alene peoples, and that this research carried out in the Sky Islands is carried out on the lands of the Apache and O'odham peoples. And this is a view from the Pinaleño Mountains, showing that as a nice picture, a nice way to show that these islands, these mountains rise up from the desert down below. And so the Madrean archipelago, or Sky Islands, is about 65 mountain ranges that form a kind of a stepping stone between the Sierra Madre in Mexico and the Rocky Mountain and the Colorado Plateau in the United States. And so while Sky Islands, the idea of Sky Islands, are not unique on the planet, this region is unique in that there's no other place on the planet that has as many mountain ranges that are Sky Islands. And so you can see there's roughly equal numbers on either side of the border. There's about 65, depending on how you define them. And these are isolated mountain ranges which rise up out of the landscape like green islands in a desert sea. I thought it was a nice quote from Brisca and Moore. And it's this biodiversity hotspot that sits at the intersection between two major deserts, the Sonoran and the Chihuahuan, and then two major cordillera, like I said, the Sierra Madre and the Rocky Mountains, which makes it really kind of a spectacular place for biodiversity on the planet. And a number of the organisms on these islands end up being isolated due to genetic drift between the Sky Islands. And so you see that there's this desert and it progresses as the biomes change as you go up the mountains. And so one of the things that makes this habitat unique, this biodiversity hotspot unique, is the oak woodland habitat. And so these are some different images from some of the different Sky Islands, just to get an idea of what it looks like. So one way to think about this, and this is, not unique to the Sky Islands, but for every 1,000 feet of elevation gain, the temperature drops around 4 degrees and annual precipitation increases by about 4.5 inches, which is the equivalent of driving 300 miles to the north. And so as these tarantulas moved into these habitats, and then they decided to move up the mountains, the magic of speciation occurs as they adapt to differing habitats. They move up into the oak grasslands, oak woodlands, the pine oak woodlands, and then all the way up into the mixed conifer forests on top in the highest elevations. And where other lineages then take off and go back into the desert. And you can think of this as you drive up these mountains, it's analogous to driving from Sonora to Canada, which is, if anyone's ever done that, it's an incredibly different, there are a number of different types of habitats that you're going to be going through. And so what's happening is on these mountains is that these biomes are getting stacked on top of each other. And so here's a view of the Chiricahua Mountains, up in the Chiricahua Mountains, just to give you an idea. And then this is what it looks like down in the desert below. And so you can see that, or you can begin to see that things that are going to be living up in these mountains are not going to enjoy the types of habitats down in the desert below. And so they're not going to be crossing these seas of desert habitat. So why are we working on Aphonopelma? Well, besides the fact that, you know, they're my favorite group, I've worked on them for a long time now. You know, there was this idea that they were, you know, perhaps one of the greatest known challenges to species delimitation in spiders. I don't necessarily think that's true anymore. I mean, I think that they're a difficult case, but I think a lot of spiders are difficult when it comes to species delimitation, obviously. And then when we started this work, there were 55 species, described species in the United States, and that the problem was that most of these were described off of only one or two specimens. And people didn't have a really good understanding of the amount of variation that exists in these traditionally used morphological characters. And so I like to show this. Some people have seen some of these slides before, and I like to show this because it's a good test. So this is for everyone that's out there sitting, if you were to see these bulbs under the microscope, how many species would you say that this is? So this is 3 species. And if you were to see these spermatheca under the scope, how many species would you say that this is? And this is 1 species. So this is the variation that can be found. And it's tremendous. And so what did we do? We used phylogenomics. Well, we used CO1 to start, and we used some phylogenomics to help us understand some of the backbone phylogeny that we could then use to reciprocally illuminate morphological characters and other pieces of information to help us understand and identify the species boundaries. And so now we have this really good understanding, at least on the North American and the US side, of kind of where some of the species boundaries should be. We feel pretty confident about the species here in the US, and that one of the things that we did is we went from 55 species down to 29 species. So a number of species got synonymized, but there were also a number of new species. And ways that we did this was looking at these traditional measurements, and then looking at not just the measurements, but also ratios to see if there were differences that could be teased apart between certain species. One of the other things that we did was look at principal component morphospace. And, you know, this is interesting too, because it's a little bit a different way of looking at your data, and you're kind of looking for clouds of morphospace. But, you know, one of the problems with the traditional view of this is that it's in a 2-dimensional space. And so some of these things that you're seeing that separate out are easily seen, but then some of them might not be seen that they can separate out because they might be on top of each other. But when you add a 3rd dimension to it, and you can see this kind of cloud, this space that they're occupying, then you can kind of get an idea. And it helps to, you know, it's not so much that you're going to use this to define the boundaries, but it's going to help to understand potentially where some of the boundaries are, and then you can go and look for other characters or other pieces of information. And the other thing too that we found was that, you know, from an evolutionary standpoint, these major species groups all occupied different phylogenetic morphospace as well. So you could see that they were on they were definitely on their own evolutionary trajectories and had shared evolutionary history. One of the other pieces of data that we used were locality data. And that was one of the things we found that was incredibly important, was where the specimen was actually from, as well as niche models to see if they were occupying different niches, potentially if they were in similar areas. And then lastly, one of the really important things is behavior, and in particular, mating activity patterns, when the mating seasons were for these, so when the males were out wandering. And so as we kind of dove back into the Sky Islands group, the Marxi species group, at the time of the revision, these were the mountain ranges that we had sampled, the mountain ranges and the species that we had sampled and described or redescribed. And so here, and this is just on the US side. And so you can see that there were a lot of them that needed to be investigated. Now, as of December, we've sampled all of these groups here in all of these mountain ranges in the United States, as well as one in Mexico. And we're just beginning to get onto that side. And one of the things that Brent found was that when we had some samples of things from the Mexico side, from Sonora, that not surprisingly, there was a new species. And this was Aphonopelma bacadehuachi that Brent described earlier this year, last year, sorry. So now we're starting to sample, and we're starting to get an idea of where these things are found. And they're incredibly difficult to find. These habitats can be very harsh, very difficult terrain. Their burrows are incredibly cryptic a lot of times, trying to find them. But now we're starting to get a better idea of when we need to be looking and where we need to be looking. And so one of the cool things we're finding is that is not just that there's one new species or potential new species on these mountains, but if the mountain gets high enough into this conifer forest, there's oftentimes another species. And so this is something we're working on now. We're working on the description. This is a new species in one of these, one particular mountain range in the Sky Islands. We looked at CO1, we looked at ITS, we looked at morphology, we looked at behavior. We started to find these things up top on the mountain, and we see that, oh man, wow, they really are. They're different. Okay, so there's morphology, there's behavior, there's molecular differences. And so now we're getting into this, sampling these other mountains, and we're getting preliminary data back, and we're seeing, all right, is this really going to hold up? Are we going to have on every single one of these isolated mountain ranges, is this going to be a new species? And you can see too that a lot of these things a lot of the Sky Island species are relatively small compared to other Aphonopelma. But there's a mix in these. We also have larger species too, not just the little ones. We have at least potentially 3 new larger species in the Sky Islands as well so far that we're working on. And based on these previous findings and some of this preliminary data, and then we look at this idea that there's this conservative estimate that there are 40 isolated mountain ranges in the United States and Mexico. There might be around 65 defined Sky Islands, but whether they're actually isolated from each other or not, or whether there was dispersal and migration back and forth, that it's not really well known yet. But if they really are 40, there's this potential for this huge number of endemics throughout this diversity hotspot. Obviously, as the dude says, this is all up to how you define species. But that's our job. That's our job to be the ones to determine that. And why is that important? Well, one reason is research is showing that there have been dramatic responses to climate change for taxa in the American Southwest, particularly in these Sky Islands, and that you're starting to, when people start to look at this, they can see that plant distributions have significantly shifted where they are found on the mountain from now versus the past, whether you're using historical collection records. And these things are moving further and further up the mountain as the climate warms. And so the animals that live in these, alongside these plants are also getting pushed up and potentially off of the mountains. And basically all of the assessments for how climate change is going to affect the Sky Islands have been based on plants and vertebrates. Not surprising, but yeah, so much of the literature and the research has focused on the US side, which is another important fact, which is completely disregarded Mexico, Western bias and such. But this is important because this Mexico side is potentially slightly larger, has more mountain ranges, potentially more diversity. And so, we feel strongly that this group of megalomorphs, we know that megalomorphs are good indicators of biogeography and understanding biogeography. And we really think that this species group, can help us understand how climate change has and will affect speciation pattern and process, as well as inform island biogeography. And so with that, I have to thank some of my friends and colleagues like Oscar and Jorge and Danny, and obviously Alex, who has not just invited me, but has also, all these people have collected specimens for us and helped us start to get to work on the Mexico side. So if anybody has any questions, I would be happy to answer them.